Alright, so before we talk about mitosis and cell cycle, then perhaps we should look at the relationship between nucleus, chromosome, DNA, and the nucleotide. Now, as we know, nucleotide is actually a basic unit of DNA. So if many of them join together, then we are going to get a molecule of polynucleotide. Now, there are two types of polynucleotides. Okay, they can actually exist in a double-stranded structure known as DNA, or just a single-stranded structure known as RNA. So in this diagram, as we can see here, now this molecule is made up of two strands. So it must be a molecule of DNA. All right, and then these two strands are actually held together by the hydrogen bonds exist between them. Now, so if we zoom out further, then we will discover that actually the DNA is a very thin, fine thread. Now, how do you organize the long threads of DNA in our tiny nucleus? Now, we need to organize them so that we are able to manage them, organize them in a more manageable way. How? It's by coiling them around the histone proteins. All right, now so we have this is actually a group of histone proteins. And when they coil together, then we get the chromatids. All right, so they are in the uncoiled state. Now during prophase 1 or prophase 2, we know that chromosomes condense and become visible. Now how is it done? It is done by the compaction. All the DNA together with the histone proteins, they coil themselves and compact themselves to form a condensed form of something known as chromosome. Okay, so let's look at cell cycle. So this is a cell cycle. A cell cycle starts with a G1 phase. When a cell is newly formed, the cell should then undergo G1 phase first. Now this is when the cell grows in size, synthesizes more organelles and proteins. Now, so by right, as I mentioned just now, chromosomes start to condense during prophase 1 or prophase 2. That means to say that before prophase 1 or prophase 2, basically we won't be able to see chromosomes in G1s or G2 phase. But just to show you then we condense it so that we are able to visualize them during the G1 phase. As we know, now we have 46 chromosomes in our nucleus, or 23 pairs of homologous chromosomes. So during G1 phase, this is the number of chromosomes that we have in each and every one of our nucleus. So 2n equals to 46, so we have 46 chromosomes here. Now these two are known as homologous chromosomes because they have the same length, they carry the same genes. Now we have 22 pairs of them. Now, so these 22 pairs are known as autosomes because they are not involved in sex determination. The last pair is actually known as sex chromosomes because they are involved in sex determination. So a male would have 44 chromosomes, 44 autosomes plus XY. And a female would have 44 autosomes plus XX, and making up a total of 46 chromosomes. Then what happens during S phase? During S phase, we know that replication of DNA occurs, or we say duplication of chromosomes takes place. Now this is when this chromosome starts to make its own copy. Now, so as you can see here, now this chromosome is exactly the same as this, and they are joined together, they are attached together at the centromere. So we call them sister chromatids because they are the exact copy of each other. Now same as this. As you can see here, now initially this is the chromosome, the chromosomes that we have for the first pair of homologous chromosomes, then it makes its own copy. So right now, for the first homologous chromosomes, we have them actually existing in a pair of sister chromatids. Okay, so we have 44 homologous chromosomes. 44, we have 44 chromosomes. and XX, or 44 chromosomes and XY. Alright, now so next is G2 phase. This is when the cell continues to grow and prepares itself for cell division. How? By accumulating more ATP. Now, so remember, right now we have 2n equals to 46, but we have duplicated the number of chromosomes. Now the cell will then go into prophase 1. Now during this prophase 1, then we will be able to see the condensed form of chromosomes. Now, so they condense, so they are actually visible under a light microscope. Now this is what it looks like under a light microscope during prophase. So of course, by using computer and some software, we are able to actually arrange them to form karyotype. Now, so how do we arrange them? We actually try to pair chromosomes that have similar length. All right. Now, so during after prophase, it will be metaphase. Now this is the metaphase. Now as you can see here, we have 46 chromosomes, or 46 pairs of sister chromatids joining at the centromere. Now 46 is not a small number. So how do you align them on the metaphase plate during metaphase? Now you can't actually align them in one straight line. So that's why if you look at this diagram, now this is just a diagram to illustrate to you. Now this is the first layer, the second layer, third layer, and the fourth layer. So the sister chromatids are arranged in 4 layers of them. And then during metaphase, the spindle fibers are fully formed. And where do they attach to? They attach to the centromere. So next, right after this will be anaphase. Now during anaphase, due to the shortening of spindle fiber, now the spindle fibers actually contract, so pulling the sister chromatids apart, forming new chromosomes that we call daughter chromosome. So during anaphase, by right, this is what you should observe now. So this chromosome, this is a chromosome, and this is another pair of. These two used to be a pair of sister chromatids. Now, due to the shortening of spindle fibers, and then they will pull apart, forming 2 daughter chromosomes. Now, what are daughter chromosomes? Daughter chromosomes are newly formed chromosomes. Basically, they are just chromosomes. All right. Now, so after that, telophase takes place, and we are able to get 2 cells after cytokinesis splits the cell into 2. Now, so if you still remember, now first of all, we have a parent cell with 2n equals to 46. So this is what we have. And then after S phase, we have duplicated the number of chromosomes. Now, after a complete cycle of mitosis, we get back 2 identical cells. Each are actually identical to the parent cell. Now remember, if you join up these 2 diagrams together, basically what do you get? You actually get them in the duplicated form, if you still remember. But during anaphase, if you still remember, during anaphase, these 2 separate. One goes to the north, one goes to the south. So that's why we have the 2 diagrams like this. Now, so each daughter cell will have 2n equals to 46.